Okay, good afternoon, everyone. Okay, so I'm back, and so we're going to start Module 3 today, and we're going to go through a number of things having to do with energy and reactions. So today we're going to start this set of PowerPoints that I prepared that correspond to your Chapter 13 in the textbook. So we're going to go through some of the basics of this idea of bioenergetics this time, and then the redox reactions next time. Okay, and then we'll actually talk through a bunch of these different pathways and everything. And just like last time where I said, yes, you need to know the amino acid structures, I will tell you right away that you need to know a lot of structures, because we're going to be talking about mechanisms and pathways and reactions and things like that. So one of the things, if you're not so great at memorizing structures, and a lot of us aren't, you might want to start looking at some of the glycolysis pathway this week, actually. Okay, as we go through glycolysis, you start with glucose, and there are 10 transformations. So by learning the transformations and the names of the enzymes and things like that, hopefully you would know glycolysis. But for some people, it kind of helps if you practice drawing the different steps ahead of time. So I'll tell you right ahead here that we do expect you to know glycolysis and citric acid cycle and some of these other pathways. So I'll give you the heads up that you can actually start looking that over today. Okay, but for this week, we're actually going to be talking about some of the background information, some of the definitions and terms and concepts having to do with this bioenergetics. Okay. Okay, so I mentioned that this set of slides that I made for the PowerPoint, I made it so that it kind of aligns pretty well with Chapter 13, but note in the information and everything, there's a little bit of information before Chapter 13. So we're starting this whole new module, and there's some definitions, some concepts. So there's a few pages that talk about the introduction to metabolism. So definitely go through that. Look over those concepts, get to know those definitions and everything. So it's the introduction to metabolism. So what are we talking about here? We're talking about a whole bunch of coordinated cellular activities. So these are all the different things a cell does. Everything from catalyzing a chemical reaction, transporting things, breaking down other things, building stuff, whatever. And we'll talk somewhat as we go through this module also about the idea that they're coordinated and regulated. Okay? And as we go about doing things like making things or breaking things down, we also need to be able to have some energy. Okay? So we get energy by breaking things down. We use energy when we build things up. Okay, and so when we think about the cell, what does the cell need? Okay, we need some of that energy, and we also need certain other materials. In particular, we need a lot of carbon. Think back on all those different molecules we've talked about, starting in like the third lecture. I guess the second lecture was water, so there's no carbon in water, but almost every other molecule we talk about, right, we're carbon-based life forms, there's carbon in it. So we need a source of carbon. And then a lot of those other compounds, right, your amino acids, your nucleotides, a lot of these compounds also have nitrogen. So we also need a source of nitrogen. I won't go into that as much, but we'll, but you can think about it as, hey, we need carbon, we need to get it from somewhere, it needs to be built up or broken down into other things. Okay, so let's go through a few definitions. Autotrophs are organisms that can get all the carbon they need from carbon dioxide. Okay, the carbon dioxide floating around in our air, right, causing our global climate change and everything. Well, autotrophs can suck up that carbon dioxide and make other molecules with it. like maybe acetate or sucrose or fructose or whatever. So that's not us, unfortunately. We can't do that, okay? But certain other organisms can. Like what? What can take carbon dioxide out of our air and make materials, carbon-based materials? What? Plants, yeah, plants, and some other, some types of bacteria too, so cyanobacteria and a few other things. But the biggie is green plants, okay? So we're not autotrophs, okay, but so we can't just suck up carbon dioxide and use that for our carbon source, but we can get carbon from plants, okay? So something that used photosynthesis and got carbon from carbon dioxide. So we can get our carbon from plants, or we can get our carbon from, so basically we can eat them, or we can eat something that ate plants. So we could eat other animals or fungi or things like that. Okay, so we're heterotrophs, okay, and plants and a few other species are autotrophs, or a few other types of trees. And so basically we have autotrophs in sunlight. They can take that carbon dioxide and they can make organic products, so small molecules with carbon in them, and then combine those to make bigger molecules, and they give off oxygen. And then we take in the various organic carbon-containing compounds, and we breathe in the oxygen, and we give off carbon dioxide. Okay, so in our own small way, we contribute to global climate change as well, but anyway, more from burning fossil fuels. But we actually emit carbon dioxide too. Okay, so we have some general terms for these various processes then of building things up and breaking things down. So in order to get some of that carbon from plants and energy and things like that, we use processes called catabolism. So catabolism is sort of the group of processes that does that. So it's breakdown of compounds with the release of energy. And it's over here in the red. Okay, so over here in red we have things like carbohydrates that we might eat, fats, proteins. So in the process of catabolism, we break them down into smaller molecules. First, maybe amino acids and lipids and everything, but eventually, if we go all the way through catabolism, we end up with carbon dioxide, water, ammonia, some very small molecules. In that process, in our metabolism, while we're doing that, we capture some of the chemical energy in these molecules over here. So ATP, NADH, NADPH, FADH2. We'll talk about what these are and how they work in this set of lectures. Okay, so we capture that chemical energy. And then we can use that if we want to take some of these precursors like amino acids, sugars, fatty acids, bases, right? And build up the macromolecules in our cell, proteins, polysaccharides, lipids, nucleic acids. Okay, so this process then is called anabolism. We're building compounds from simple precursors, and we need to put in energy. Okay, so we're going to talk about how we do some of this breakdown, how we do some of this buildup, and how does that energy play a part? Okay, how do we get it as we break something down, but also how do we use these sorts of molecules when we want to build something up? Okay, and the sort of overall way that a lot of these things are done is through steps in biochemical pathways, each catalyzed by usually an enzyme. Okay, so we can have something like, here's glycogen broken down to the molecule glucose, that might be broken down to the molecule pyruvate, that is broken down to acetyl-CoA, and then further. Okay, so we can have this pathway where there's an enzyme that takes glycogen and makes glucose, or more often a series of enzymes. And we can go all along here with a whole pathway, and it can be linear. You can go from glycogen to glucose, pyruvate, or whatever. Okay, but our cells are actually much more complex than that. And we have other pathways that can also lead to this molecule of acetyl-CoA. So you can also take phospholipids, break them down to fatty acids, and get that. Okay? And then there's also some of these amino acids are feeding in over on the side here, for example. So we can have convergent pathways. So you can use different things. You can eat a lot of different things and get energy from them. Okay? And they come down here, and for example, the glycogen is broken down to glucose, sucrose broken down to glucose. Once you get the glucose, goes down the same pathway to get to the acetyl-CoA. Okay. Coming out of that, well, you can use a molecule to build another molecule, and then sometimes you have a choice of different things you can make. So it can also be divergent. You can make a bunch of different things from one precursor as well. Then you can also have cyclic. Okay, so we're going to talk about the citric acid cycle in a few weeks. I guess it's after spring break, right? So you can take a compound, feed it into a pathway, things happen, and you come around and you actually have one of the substrates is recreated and feeds back into the pathway again. So there's all kinds of different pathways, and we can eat lots and lots of different things to get the precursors that we need and the energy that we need. And from a bunch of relatively simple molecules, we can make all kinds of different things in these divergent pathways. But another thing to think about, I kind of wrote out over here, is as you look at these pathways, you say, well, you know, there's different ways we can make the same thing, and there's different ways we can use certain molecules. So we might want to have some sort of organization. We don't want everything going on at the same time, right? If you have like a glucose molecule, maybe you need to make some sort of carbohydrates on the cell surface, but then again, maybe you need to break it down to get some energy, or maybe you want to use it over here to make some 5-carbon sugars for making DNA or something. So you need to have organization and regulation. And there's some ways of doing that. So you might have different enzymes, right? So in these pathways, you might have a different set of enzymes that's going to take this molecule and go up here versus down here. Okay, that makes sense. You might even have different versions of the same enzyme, like perhaps in different cell types or different types during development. You also have different locations, right? You have different cell types, again. You have different parts of the cell. Remember way back in that first sort of introductory chapter, we talked a little bit about this idea that, hey, you have these different organelles in eukaryotes. And even in bacteria, you have different regions of the cell. You might have different things like a flagellum, or you might have an area around the DNA. You might have other groups of proteins in different places. So you have different locations, so you can have some pathways happening in one place and other pathways maybe using some of the same compounds in another part of the cell or in another cell type. There might also be, availability of substrate. Well, you're not going to break down glucose if you don't have any glucose to break down, for example, right? So there might be some regulation in just making available some of the molecules at the right time for the cell to use them. We also have allosteric regulation. I think you went through a little bit of that when you talked about enzymes last module. The idea that you have different signaling molecules or other proteins that can come in and bind to an enzyme and turn up or down its activity to help, again, direct which pathways or parts of pathways should be on or off at different times. And then finally, something we don't go into a lot in this class, but we have these overall regulators of growth factors or hormones that certain cell types might make, send out often in the bloodstream, and they go and affect a whole bunch of different cell types or a whole bunch of different genes within a cell, turn on whole sets of gene expression, which means they turn on the making of the enzymes that do some of these pathways. So at different times of the day over the year, whatever, you might have different sets of enzymes made in certain cells. Okay, again, it might also be different times of development of a certain cell. Like I was just talking to somebody about blood cells. You have stem cells, and they divide, and then you get red cells and white cells, and different types of white cells, and the B cells might sit there until they're triggered to become antibody-making cells and things like that. So at different times and different places in their development, they might be making different sets of enzymes, and some of that is regulated by these growth factors or hormones that some cells will make. And please limit your conversation. I know even a little bit of very quiet talking, other people definitely hear. It really echoes in here. So please hold your conversation till later. Okay, so look over those few pages in the textbook before chapter 13. I went through some of the definitions here and everything. There's a little more in there to go through also. Some of it might be a little bit of reminders from other classes, but definitely go through it. And now we can go through some more in-depth this idea of bioenergetics. Okay, so again, if we're going to be making up these complex molecules like DNA and proteins, everything, we need energy. Okay? We also need a number of other things for survival that take energy. We need concentration gradients. Okay? If you didn't have different concentration of certain ions on one side, like outside your nerve cell and inside your nerve cell, you wouldn't be alive, basically. You wouldn't have any neuronal signaling. You wouldn't be able to respond to anything or whatever. So you have to have concentration gradients. You have to have different concentrations of certain molecules in different places. And often we think of it as, you can sort of picture this on one side of the membrane, so the cell membrane, or the other. But it could also be inside the nucleus versus outside, or even just other regions in the cell. Okay, and so some of that I kind of mentioned with that is this idea of electrical gradients, charged molecules like ions on one side of a membrane versus another. Okay, motion. We talked a little bit about muscles, I think, last time I was here. So in order to move, we, of course, need that ATP molecule for our muscles. There's other types of movement. There's other types of proteins that move around within our cells or between cells or our whole organism, or bacteria with their flagella or cilia or whatever. So there's different types of motion. And heat, okay, when we shiver to get warm and everything, we need heat. And this last one, we don't really depend on this too much, but it's kind of cool anyway, is light. Okay, so we don't really, literally, we don't really glow like some of these other organisms. We might have a glow, but we don't glow like this. So anyway, I don't know if any of you have caught, we call these lightning bugs, okay? But I guess the real term is fireflies. But back in Ohio, we called these lightning bugs. So these are a type of beetle, and usually you can find them kind of around sunset out in the backyard. They just kind of fly around slowly. They kind of hover in the air. And the abdomen lights up. So if you're standing quietly in the backyard in the summer, you see these little bursts of light. They go on and then off. And if you walk over carefully, you can often just kind of collect them right out of the air, and they'll kind of glow in your hand there. So that was something we did as kids, is collect the lightning bugs. So that's signaling, trying to meet up with lightning bugs of the opposite sex, basically. Deep sea organisms also, some of them create light as well. And the deep sea organisms, in some cases, they use it for actually attracting prey. Okay, so they to help them get something to come over so they can eat it. Okay, so we don't really produce light, but it is very important for a number of different organisms, and it does require energy to do that. Now, for the most part, where does that energy come from? Well, it comes from the sun, okay? Our star, the sun. Okay, so the ultimate source of energy for most things is going to be that sunlight. And the bioenergetics that we're talking about is how do we go from sunlight to something we can actually use and then use it, okay? And we're actually, we're going to kind of skip that first step, okay? How does it go from sunlight to, we're not going to do photosynthesis, but we're going to talk about how we might get molecules from other species, like from plants or whatever, in this course, how we can get energy from molecules, convert it to a useful form, and then use it. Okay, as we go through this section of lectures, how do we get energy, where does it come from, right? How do we use it and everything? Even though we're talking about living things, this is not some magical thing that's different from other physics, okay? This is still physics, biophysics. We're still going to obey the law, that is the law of thermodynamics, okay? So the first law is the conservation of energy. Hopefully you remember this from physics. Okay, I don't remember. I don't know if you remember or not, but if you don't, you can go back and look over it, and again, the book goes it reminds you very well. So the conservation of energy in the entire universe, the total amount of energy is constant. It may change form, okay? You can transport it, but you're not actually creating or destroying it, okay? So we're going to get it from somewhere, it's going to go somewhere, okay? The other thing, the second law that we're going to go over here, we're just going over two of the laws today. The universe tends towards increasing disorder, okay? If you see my office at home, this is pretty obvious, okay? It's very easy for papers to end up all over my desk, on the floor, on the spare bed, everywhere, okay? So it's very easy for things to get in disarray. And then it takes energy to go put those things in order, okay? So, well, I gave you the answer here. So basically, it's very easy for things to go towards disorder. Okay, increasing entropy, you might remember from physics class. So how do we do things like make something as complex as a protein or DNA or something? We're going to put in energy. Okay, so just like it takes energy to clean up all those papers, journal articles in my office, it takes energy to make DNA, proteins, carbohydrates, other molecules. And what essentially is happening there, so we're not breaking the second law of thermodynamics, is we're getting that energy from somewhere, and that energy, when it was put into whatever form that is, results in decreasing order somewhere else. Okay, so we're we're increasing entropy, we're increasing disorder because we're getting some energy from something. And in this little local environment where we're making complex things like DNA or proteins or something, we may be increasing order, but elsewhere there's decreasing order. Okay, so in order to talk about how all this happens, this input of energy and where it comes from and things like that, we have a whole bunch of definitions. Okay, it's not too many, but you know, I've kind of written out the key concepts in these next few slides again. So let's go through them in order here. So G, Gibbs free energy. Okay, so we're going to talk about free energy, and it has a definition of the amount of energy capable of doing work in a reaction. So we'll talk about changes in free energy. Okay, so changes in G, delta G. Okay. But the free energy is the amount of energy capable of doing work in a reaction, and technically it's at constant temperature and pressure, And we usually write it in joules per mole, these are the units, or calories per mole. When we have a reaction occur, and we've given off some free energy, we've released some free energy, we say that the change in free energy, or the delta G, is negative, and it's an exergonic reaction, so it's exo, giving out, okay? If we need some energy to get that going, if we're going to build up bigger molecules or something, and you've got to put in some energy, so we say that the change in free energy, or the delta G, is positive, and it's an endergonic. So ender, think of it as putting in energy. So if we're thinking about, oh, is this reaction likely to happen? Is it going to occur? Well, think of it as, well, it's probably going to be exergonic, giving off energy, as opposed to needing energy to make it go, right? So if it's a spontaneously reacting system, it's going to be a negative free energy, if we're talking about a particular reaction, okay? And then I talked a little bit about this idea of entropy. It's a way of expressing the randomness or disorder in a system, okay? And when you have a gain in entropy, you actually have more disorder, more mess, okay? And then, so the entropy of the universe, whatever we're doing, whatever we're building up, if you think of the universe as a whole, it's always increasing. But in a reacting system, so in a little area where we're talking about a cell or an area or a test tube or whatever, entropy can be decreasing. So we can be building up things, okay? So overall, as we talk about biological systems and living organisms and everything, we're going to get free energy from the environment. Again, if we were plants, we could get it from the sunlight, but since we're not, we're going to get it from nutrients or eating stuff, okay? We're going to eat stuff, we're going to get that free energy, and we're going to return an increase in entropy and also some heat, okay? Okay, so what we can do as biochemists or biophysicists is think about some of these different processes in the cells and in the organisms, and think about, well, how much free energy might we need to do a certain thing? Okay, how much free energy would a particular reaction give off? Can we connect those somehow? Something giving off free energy, something taking it, anything. So we can do all these calculations about changes in free energy for different reactions. So we can predict the direction of chemical reaction. Is it likely to go from A to B? Or does that require a lot of input of energy? So maybe it would actually be easier to go from B to A. Okay, so the direction of a reaction. Determine the equilibrium position. We'll talk about what equilibrium is, but let's, it's kind of like that point of balance, I guess is how I tend to think of it. And then we won't go through this a lot in this class because of the sort of short nature of this one semester biochemistry class. But when you know about how much free energy is given off in a procedure, you can also use this sort of information to figure out just how much work that process can do. How much, motion can something do? How big of a package can somebody pick up? Or, how many proteins can be made in a system? Okay, so you can actually think about the amount of work that can be done. We're not necessarily going to go through the details of that in this particular class, though. Okay, so let's go through a few more definitions we need before we can talk about specifically some of these reactions. And one is delta G 0. See, there's a little 0 up there. So when we talk about free energy changes in biology, we sometimes use this delta G 0, or even down here, delta G prime 0. So delta G 0 is the standard free energy change. meaning the free energy change at standard conditions, and that's a definition that's defined as 298 Kelvin, which is 25 degrees Celsius, okay? All the reactants and products, okay, so each reactant, each product is a 1 molar concentration. And then gases like your carbon dioxide, we mentioned, the oxygen, right, things like that, any of the gases in the reaction are at 1 atmosphere. Okay? Now, in biology, a lot of things that are going on in cells and organisms and everything, right? We're not necessarily going to have everything at 1 molar concentration, right? That doesn't really make sense in biology, everything at 1 molar concentration, right? So we have what's called standard transformed constants, so delta G prime 0, and something called K prime equilibrium for biological reactions. So here we're going to add in this idea, this part of the definition, pH 7. Okay, so unless otherwise noted, we're going to talk about pH 7, so delta G prime 0 at pH 7. We're going to have some water around, because a lot of these reactions we're talking about are going to be involving water, or going to be in water. They're in aqueous solution. Not all of them, but a lot of them in biology. So 55.5 molar. We're going to have a little bit of magnesium around. That's needed by a lot of enzymes that we're going to be talking about, so there's a little bit of magnesium. And another thing that some of you already noted earlier in this class is we often don't write down specifically how many water molecules or protons or magnesiums might be in that particular procedure. I know, like in chemistry class or organic chemistry class, you really count everything and make sure every hydrogen atom and every carbon, well, we do worry about the carbons, but every hydrogen atom, every proton, everything, every water, you want to balance the equation very, very, very carefully. And sometimes you'll see more in these biology reactions, we talk more about, hey, here's the glucose, and then here's the pyruvate and stuff. We don't necessarily count every proton and everything. So you might notice that's a little bit different in some of these biology reactions or biochemistry reactions. Okay, a few more definitions here. So one of the things that we often think about is this idea of equilibrium. So when we have a particular system, like a cell or something in a test tube, and we have some enzymes and we have the reaction going, the reactants and products change. You know, the enzyme takes the substrate, makes a product, and maybe might take product and convert it back to substrate and everything. There's a certain amount of reversibility. At some point you reach what we call equilibrium. And that is the point where you have reactants and you have products, and the rates of going forward through that enzyme and backward are equal, so that if you're just looking out here at the test tube, it looks like the amount of substrate is not changing, the amount of product is not changing. Okay? The reaction might still be occurring. You might still be having some substrate going to product and some product going to substrate, but it's equal, it's balanced out. Okay, so you have this point where you have equilibrium. And we can talk about what that point is for a particular reaction by using something called the K equilibrium, equilibrium constant, the ratio of the reactants and the products at equilibrium. Okay, so this is our our way of writing like a example equation here. Some small a number of molecules of molecule A, some number small b of molecule B gets converted to C of molecule C and D of molecule D. Okay, so the small letters are numbers, the big letters are molecules. And this is our K equilibrium. So up here in the numerator, we have the concentration of molecule C. So it's a concentration, like a number of moles, right? 2 moles, 3 moles, whatever. The concentration, moles like moles per liter, right? Think of concentration. To the small c power, concentration of D to the small d power over the concentration of A to the small a power and the concentration of B to the small b power. And of course you can do this with, even if you have just one substrate and one product, or a whole bunch of substrates and a whole bunch of products, you can calculate this K equilibrium. So again, the numerator is the products when your equation is written in this order, and the substrates go in the denominator. Okay, so we can do this calculation of what is the K equilibrium for a lot of different reactions. And the other thing about this concept of equilibrium is that it's what drives a lot of reactions. So things are going to tend to go towards equilibrium. So you have substrate and you have product and a test tube and you have an enzyme, and you've got substrate going to product and some product going to substrate and everything. It's going to tend towards getting to that point where it's in equilibrium. And this force of getting to equilibrium is related to that delta G that we just talked about a little bit, the free energy change. Okay, in fact, we can relate the delta G prime 0, the free energy change of a reaction, to the K prime equilibrium. Okay, there's some other stuff here. Okay, so let's think about this first. So we got delta G prime 0, we got K prime equilibrium. Okay, this is natural log. Okay, one of those buttons on your calculator that you don't use very often. Okay, this is a natural log of K prime equilibrium. T, what's T? Guess? Temperature, okay. Yeah, very good, thank you. Kelvin, yeah. And R. Okay, so the gas constant is R, 8.315 joules per mole K. That's your Kelvin again, okay. Or if you have kilojoules, okay, 0.008315 kilojoules per mole K. Now, if we're doing our little reaction in the tester, right, sitting there, you're going to have a particular temperature, room temperature, or if maybe you have in the water bath or in the incubator or something like that, right? There's a temperature, right? So that's a constant. And then R is this number, so it's a constant. So that means that the free energy change depends on the K equilibrium constant. Okay? So you can, if I say, oh, here's some concentrations of substrates and products for a particular reaction at equilibrium. Oh, you can plug it into this, I can't point at the screen here, right? Plug it into this equation here and get your K equilibrium, your equilibrium constant. Plug it into this equation here, and you can figure out what the free energy change of that reaction is. Okay? And over here in this chart, this shows how free energy change and K equilibrium, how they relate to each other. And it's slightly confusing because they did it twice, okay? So this is just kilojoules per mole and this is kilocalories per mole. But in either case, you can say like, oh, look, if my K prime equilibrium is really big and positive, my free energy change is negative, okay? Whatever units you use, whether it's kcals or whether kilojoules, okay? So does that make sense? So let's see. So big here, lots over here. So the free energy change is going to tend to go towards. So the reaction is going to tend to go forward. Okay, and the free energy change is negative, which means the reaction is going to tend to go forward. Okay. If you have a very, very small number, remember like 10 to the minus 1, 10 to the minus 4. These are very small numbers, right? Very, very small numbers. Plug that into K prime equilibrium. It's going to get, the natural log of that's going to be negative. Oh, but over here you have another negative, so now that becomes positive. The delta G prime 0 is now positive. Okay? And if you kind of compare this equation again to this, so we're saying if your K prime equilibrium is very small, oops, back up. Okay, very small. That means this is the numerator is small, denominator is big, so you have more here than here, so that means you're going to tend to not go forward in this reaction. You're going to tend to go the other way. And that makes sense because then delta G is positive, so it's an endogalvanic reaction. So if you go back to these slides and think about it, combine it, these are not separate different pieces of information. This all kind of goes together. You've got your delta G, whether it's positive or negative. You've got your K prime equilibrium, whether it's big or small and everything. And then you can kind of summarize it like this. You got K prime equilibrium, bigger than 1, negative. Delta G prime 0 is negative and proceeds forward. Okay, it's exergonic. K prime equilibrium is less than 1. Think about what that means for those different equations. Delta G prime 0 is positive. Could actually proceed in reverse. When you get to 1, if K prime equilibrium is 1, what if we get 1 here? Well, you could take the natural log of 1, you get 0. Natural log of 1 is 0, okay? So 0 times T times R is still 0. So delta G prime 0 is 0. And that means that that reaction under those conditions is at equilibrium. Okay? Okay, so we might say, okay, well, that's very nice. We can think about equilibrium, and we can think about a reaction and stuff like that. We can do some of these calculations under standard conditions, okay? But in our cell, we don't have standard conditions. Remember, go back here to standard conditions. What were they again? Standard conditions, 25 degrees C, okay. Everything at 1 molar concentration. Well, again, we would be dead if all our molecules were at 1 molar concentration. So we try to avoid that. Okay, so we're going to think about what could an actual free energy change be inside a cell, for example. Okay, so this is our actual free energy change. See, there's no prime or 0 up there. This is our actual free energy change. So what we can do is we can take that standard transform free energy change, maybe we get it off a chart, okay, and we can plug in this information. So now we have R and T again, okay, R is still constant, T is still whatever you measure, whether it's in the cell or in the test tube on the bench or in the water bath or whatever, and we put in this formula here. Okay, now this kind of looks like what we just had with the K equilibrium. It's not though. This is actually something different. This is Q, okay? So Q is our mass action ratio, Q. Okay, kind of looks like what we had before. It's different, okay. And this is going to be the actual concentrations, whether it's in the cell or in the test tube, whatever, of our products C and D and our reactants A and B. And here, remember before we had the 25 Celsius, 298 Kelvin? We're going to put in the actual temperature. Okay, so if it's not 25 degrees Celsius, let's say it's like 10 or something like that. We're in Chicago, so let's say it's 10 degrees or something. But so you'd want to take whatever that temperature is in Celsius, convert it to Kelvin, and or if you're in Fahrenheit, go from Fahrenheit to Celsius to Kelvin or whatever, and plug that in for T. Okay, so we can figure out the free energy change for our reaction at the actual concentrations and at our actual temperature that we're working at. Okay, so that's nice. You can do the homework problems and everything, but even more importantly to our cells is that the actual free energy change of a reaction can be negative, and the reaction can go forward spontaneously even if we have a delta G prime 0 or a standard free energy change that's positive. Okay? So positive free energy change, remember, that means we need to put in some energy to get it to go. But what we can do in the cell, sometimes because we have these different concentrations of molecules, we can actually have something occurring spontaneously, even if that standard free energy change might suggest it doesn't go spontaneously, right? So we call this the mass action. The amounts of the reactants and products can affect whether or not the reaction proceeds. And if you think about this different equations and everything, what's happening here is we're removing the product. Okay, so we're removing the product. This ratio, where we have C and D, the products, over A and B, the reactants, and all these are concentrations, and then to the power of little c and little d and little a and little b. If C and D, concentrations of C and D are very, very small because we keep taking away C and D as soon as we make it, that means this ratio Q can be below 1, okay? This over here can be below 1. And then when we have RT log, natural log of that ratio, we put all that together, that can end up being a large negative value. Add all that to our standard free energy change, and that means the actual free energy change in the cell or in the test tube, wherever we're doing it, can be negative, and then the reaction can occur spontaneously. Okay, so the actual free energy change in a process depends on, yes, the standard free energy change, but also the actual concentrations of reactants and products. Okay? And the free energy change is more favorable if the reactant's concentration exceeds its equilibrium concentration. Okay, so I jotted out some other notes to remember. When we do all these calculations, whether it's the homework, the extra other questions I gave you, or things in the back of the book, or on the exam, or whatever, we can do these calculations and say, oh, wow, I can burn up one, molecule of glucose and get all this energy and everything. What we're calculating is what's theoretically possible. We don't always get all that energy converted to something else in a useful fashion. We're always losing some of it. Okay, it's not 100% efficient. We're always losing some energy. Also, even though we're calculating these things and saying, hey, this molecule, it would be so favorable for all the sucrose in my sugar bowl to just be converted to, carbon dioxide or whatever, to be broken down. It might be very favorable. Things that are thermodynamically favorable might not proceed without sufficient activation energy. Okay, so if you have a fireplace and you have some firewood, usually it doesn't just burst into flames, right? You actually have to strike a match, okay? And you can think of that as kind of what happens in the cell, too. You don't necessarily have enough activation energy for something to just get converted to something else, even though it might be very favorable for that thing to become something else. And in the cell, that's what our enzymes do. And you just finished a module where you talked about enzymes and everything. The enzymes lower that activation energy. They help bring it down so the reaction can proceed. The enzymes do not change those equilibrium constants, though. Okay, so that's not going to be changed, just how fast the reaction occurs. Another thing about delta G that's kind of neat, really, I think, is that it doesn't matter how you get from reactants to products when you're calculating the delta G. Okay? When you're calculating the delta G, what is the change in free energy from those reactants to those products? It doesn't matter how you got there. It's only the nature, what are those reactants, what are those products, and their concentrations. How much of the reactants, how much of the products. Okay. So when we think about all these different processes in the cells and everything, there's a lot of things going on. Okay, we're not always talking about just one single enzyme catalyzed reaction. We're often talking about pathways. We're talking about groups of things that occur maybe one after the other. So we can think about things like molecule A being converted to molecule B, and then that B being used for something else. Let's convert it to molecule C now. Each of these two reactions has a standard free energy change. As we do these different calculations or talk about these different reactions and everything, we'll call them delta G sub 1 and delta G sub 2. Okay, so reaction 1, reaction 2. And what happens is we can say, well, you know, A is being converted to B, but then B gets used up and is converted to C. Overall, we go from A to C. Okay, so we have A to C, and now we have a delta G total. Delta G total, all we're doing is we're going to add up delta G1 and delta G2. Okay? Simple, great. Maybe it's an answer to your homework question, but what does that really mean? Okay? What it means to the cell is very important, because if you have something that's unfavorable, like taking amino acids and making a protein, something we really need, right, that's unfavorable. But we can get it to go anyway because we're going to connect it to another reaction that is very favorable, okay? A highly exergonic reaction. Okay, so here's an example. Here's glucose. Okay, glucose. I mentioned glycolysis, the first step in glycolysis that we'll learn about in another week or so. Okay, is you're going to attach a phosphoryl group to glucose. Okay, so you have glucose floating around in a solution, you have inorganic phosphate floating around in a solution. Kind of hard to get them to come together and get that inorganic phosphate to stick to the glucose with a covalent bond. Okay, so it's going to be a free energy change of a positive 13.8 kilojoules per mole. Okay, that's not easy to do. But look over here, if we take this molecule called ATP and chop off one of its phosphoryl groups, okay, hydrolyze it, ATP, ADP to PI, that's actually very favorable thermodynamically, negative 30.5 kilojoules per mole. Okay, so let's link those together. Let's take ATP and glucose, end up with this ADP and our glucose 6-phosphate. We just add those two together. Okay, here's our adding, add it together. And overall, to get this, delta G prime 0 is negative 16.7 kilojoules per mole. Okay, so it wasn't favorable to just take inorganic phosphate and stick it on glucose, okay? But the second thing was favorable. So overall, we have favorable reactions. So this nice little delta G total is delta G1 plus delta G2. That's very profound because we can connect reactions that need energy to reactions that give off energy. Okay. But you also notice that I didn't say how this happened. I didn't draw you an enzyme. I didn't give you a reaction and, you know, all that other stuff. Basically, I just said we're going from glucose and inorganic phosphate to glucose 6-phosphate, right? I didn't say how. So the path is not important. It's just that we have ATP and glucose, and then we get ADP plus glucose 6-phosphate, okay? The beginning and end states, the reactants, the products. So coupled reactions, if you have sequential reactions and you go from A to B and then B to C, you can do these sorts of calculations and you can get a product, even if one of those steps in the middle might not be so favorable. And there's a question, yes?
Why is it favorable for a different molecule to break down this inorganic phosphate?
We'll talk about that soon, yes. So, good question. Okay. We'll talk about ATP. I have actually several slides about it. It's a very important molecule in many, many, many reactions. So, yeah. Okay. So we can figure out delta G's if we have coupled reactions. We can also come up with some equilibrium constants. So here now we have the glucose and the inorganic phosphate again, giving us glucose 6-phosphate. Let's think about this. The K prime equilibrium, glucose 6-phosphate concentration in the supernatant to just the power 1, because there's only one molecule of it, over concentration of glucose, again, to the power of 1, and the concentration of inorganic phosphate to the power of 1. The second reaction was ATP plus water, ADP plus PI. So ADP to the first power, inorganic phosphate to the first power, over ATP to first power. Okay, and then and we're given some numbers. Okay, here are the numbers, what those are. The couple reaction, again, we're going to have our ATP with the glucose giving us ADP plus glucose 6-phosphate. And I'm sorry, I can hear people talking, and if I can hear you several rows away, believe me, everyone else can hear you too. So please, please, please hold your conversation till later. Yes?
You had mentioned that we don't use H2O for calculating the AQ.
Right.
And it also mentioned magnesium, so if we had magnesium in the same equation, say, whatever it is, we wouldn't include magnesium in the KEQ?
Right, so magnesium, right. You don't use the protons and everything. So in most reactions, you wouldn't use the magnesium and everything. I'm just trying to think if there was like any chemical reaction where you would actually be making a bond or something, but no. So usually the magnesium is usually there to help you hold the ATP or the ADP into the active site of the reaction. So it's not actually so it's involved in the mechanism, but it actually is not becoming part of the substrate or the product. So. Okay, and you're right. We're not worrying about our waters and our protons when we figure out these concentrations. Okay, for the coupled reactions, okay, we have the ATP and glucose, ADP plus glucose 6-phosphate. Again, we put our supernatant over here. We take our products and our supernatant. Each of these is to just the first power, and then ATP plus glucose, again, in the denominator, and each of those concentrations is just to the first power. But if you look at this, what is that? That's equivalent to K prime equilibrium 1 times K prime equilibrium 2, right? So you've got, here's K prime equilibrium 1 and K prime equilibrium 2 over here. So you multiply those together from these numbers up above, and you get this number, and that should be raised to the power. That should be a power right there. Okay, so great, we got a lot of numbers. Now you can do some homework problems, but what does it really mean? Okay, so by coupling ATP hydrolysis, so we're breaking down ATP, to making glucose 6-phosphate, the K prime equilibrium has been raised by about 2 times 10 to the negative 5. So we went up from here, right, that's a negative 3 to 10 to the 2. So we're going to be able to make a lot more glucose 6-phosphate. Okay, so we've changed the equilibrium overall, or overall, I guess we have a different equilibrium when we combine it all together, and we have, it's favorable from over here where we have the addition of the delta G's. Okay, and now we come to the question about what is ATP doing? Okay, why is ATP helpful? ATP we're going to see in so many of these different reactions. So we're going to have a few slides here about how is this hydrolysis of ATP so favorable, okay? So this is what ATP is. I think you've seen it in some of the previous lectures in this course or in other courses. So we have an adenine base, a ribose sugar, and then we have these 3 phosphoryl groups. Okay, so this is adenosine triphosphate, right? 1, 2, 3 for triphosphate. And when we hydrolyze this, here's our water coming in attacking the gamma phosphoryl group, and that comes off as that inorganic phosphate. And then down here we have adenosine diphosphate, 2 phosphoryl groups. Okay, so why is this so favorable? Well, this is likely to go forward and less likely to go back because once you've gotten these 2 products, you've got the inorganic phosphate and the ADP, there's a number of things that these features of these molecules that mean they're not as likely to come back together. So when you have ATP, right, you have some negative charges on this gamma phosphoryl group and the beta phosphoryl group and the alpha phosphoryl group, right? There's negative charges. You separate them, right? Inorganic phosphate goes and floats away. There goes two negative charges, and then the ADP, it has two others. So bringing things together, when you've got two negative charges and two more negative charges, that's kind of hard. So the products give you better charge separation. Better salvation of products, again, you've got some charges and some partial charges and everything, and you get lots of water molecules around it, solvating it. Think back to chapter 2 there. Again, it's harder to bring things back together, so it's much more favorable to break down this ATP. Okay, and then more favorable resonance stabilization of products. Okay, if you look at the ATP, remove this inorganic phosphate, and it doesn't just stay exactly as we've drawn it there. Okay, first it's not two-dimensional; it's a nice tetrahedral shape, but it doesn't stay like that. You can get this resonance stabilization. These negative charges are actually kind of shared all the way around the molecule. So you get these partial charges. It becomes a very stable molecule floating around a solution. You also get some ionization of the ADP, so you can lose this proton. And to get this molecule to come back up here and this molecule to come back up here and bring everything back together again is going to be challenging. Okay, it's going to knock me that favorable compared to, I mean, it definitely happens. Of course, we're going to make ATP in the cell and everything, but it's not as favorable as just removing the gamma phosphoryl group and letting it float away and be resonance stabilized and everything. Okay, so this hydrolysis is very favorable, and it's coupled to many, many, many reactions in the cell. So we'll just, in many cases, we'll just talk about, oh, yeah, we add an ATP here or add an ATP there. We talked about it already, I guess, when we talked about muscle movement and things like that. So ATP, we sometimes talk about as our energy currency. We have a number of processes that make ATP, and then that molecule can, once we have a bunch of it, can be used in many, many, many other processes as well. Okay. When we talk about using ATP, then we talked about that idea of, oh, the phosphoryl group comes off and yay, free energy is released. Chemically what's happening is it's not necessarily just coming off and floating away, okay? Although that's so how we count it when we do the bioenergetics. In many cases, what's actually chemically happening in the reaction is the phosphoryl groups being handed off to another molecule. Okay, so for example, phosphorylation of glucose or other esters, or other things with hydroxyl group to make an ester. Okay, so glucose, here's most of the glucose molecule, and then here's one of the hydroxyl groups, and we attack, and this gamma phosphoryl group gets added on, and we have glucose 6-phosphate, a phosphate ester, and that happens in many, many, many types of molecules. We talked about phosphorylating amino acids, remember, but also all these different sugars and everything. Okay, so it's not, chemically in the cell, it's not just releasing a phosphoryl group and it floats away. That's how when we talk about the bioenergetics calculations, we talk about that hydrolysis being very favorable and everything. But when chemically what's happening in the reaction, it's often a handoff of part of the ATP molecule off in this phosphoryl group. Okay, and again that shows here. So if you took your glucose with a hydroxyl group or something, and you had that 18 oxygen label here, you can see that it actually picks up the gamma phosphoryl group. You can also have attack at the alpha or the beta phosphoryl groups, and you can actually transfer not just one phosphoryl group, but actually two. Okay, so that's a pyrophosphoryl group. So in that case, you'd be adding on two phosphoryl groups to this molecule. And then another thing that can also happen is some molecules, we don't talk about it often in this particular class, but if you attack at the alpha, you can also, in some reactions, pick up the sugar and the base instead. So that's called an adenylyl transfer. Okay, so when we use ATP, a lot of the cases we're going to talk about is we're removing that gamma phosphoryl group. We're either just hydrolyzing it or we're transferring that gamma phosphoryl group to another molecule. But there are other ways of using ATP and helping to promote reactions. Okay. So I was saying, like, ATP is so important. We hydrolyze as a free energy of hydrolysis. But we also have to be able to make ATP, right? We also might have other molecules we sometimes want to use to get energy. There are other molecules that are sort of what we call energy-rich compounds, or whatever. And a lot of those are other phosphorylated compounds as well. They can have a large delta G of hydrolysis. So phosphoenolpyruvate we'll talk about with glycolysis, 1,3-bisphosphoglycerate. ADP can also, in some cases, donate a phosphoryl group. So there's a range of different delta Gs, different free energy changes. Some of these can actually be used to make ATP, and then ATP can be used to phosphorylate other molecules, and you can get things lower than that in this free energy chart. When we talk about some of these other molecules, like phosphoenolpyruvate, again, some of the same factors that we talked about with the ATP here, being good at giving up a phosphoryl group. Again, you've got that electrostatic repulsion in many cases. You often have resonance of the products, and maybe also additional changes in that product. So it's kind of like modifying the product so you don't go in the reverse in the reaction. Okay, so this is our phosphoenolpyruvate. Again, here's a phosphoryl group. And in this case, once you've given up the phosphoryl group as inorganic phosphate, you get this version of the product called pyruvate in its enol form. And then you get a little bit of movement of some of the bonds of some of the electrons there, so that now you have pyruvate still, but this is in the keto form. So this tends to go in this direction. This is what you would most likely find was like floating around the cell. So it's much harder to go from here, from here back to here, add on the phosphoryl group and go here. So it's that final tautomerization or final change of the product that helps pull this reaction forward. And this actually is a very high standard free energy change of negative 61.9 kilojoules per mole. And then this is another one, bisphosphoglycerate. Again, you remove that phosphoryl group, you get 3-phosphoglyceric acid, and if you remove the proton, this ionization gives you some of that resonance stabilization. So again, this is somewhat stable, and you can think of it as just kind of harder to get this to go back here and then add on the phosphoryl group. So this again has a fairly high free energy of hydrolysis, so negative 49.3 kilojoules per mole. Okay, and this little chart's kind of nice because this just kind of illustrates the idea of you have ATP, and ATP can give its phosphoryl group to things like glucose or glycerol, or just give it off as inorganic phosphate in a solution. But there are these other molecules up here, some of which we talked about, that have an even higher free energy change of hydrolysis, and they can give their phosphoryl group to ADP to make ATP. So that's all what this is talking about. And we have sort of a somewhat arbitrary cutoff here. We think of ATP and these other molecules as high energy compounds, and other things down like glucose 6-phosphate has a phosphoryl group. It's not really going to hand it off to ATP too often or anything. Okay, so we tend to call these low energy compounds. Okay. Phosphorylated molecules aren't the only high molecules with high energy of hydrolysis, high free energy of hydrolysis. We also have thioesters, and a molecule we'll talk a lot about is acetyl-CoA. This is a donor of acyl groups, 2 carbon groups, and it feeds these 2 carbons into a lot of different metabolic pathways, but one that's very important is actually making fatty acids to make lipids and everything. And when we talk about acyl transfers, so we're not just handing this off to water again, we're actually handing it off to build up something. We're handing it off to another molecule. And this is a sort of a cartoon of acetyl-CoA because this molecule is actually very big. Okay, but this is the acetyl-CoA, and you have 2 carbons, and this would be given off then as acetic acid, and you get ionization to get to acetate. So this acetate plus the rest of this molecule, again, it's kind of hard to go backwards through this to get the acetyl-CoA again. I mean, certainly in an enzymatic reaction, it can be set up, it can be done, but it's a little more favorable to go this way, so the delta G, standard free energy of hydrolysis, is negative 31.4. Okay. And I think, oh yeah, so this is the last point I want to make here about all this bioenergetics and reactions and using these calculations and the different types of reactions and everything, is that, again, remember that we might draw it or talk about it as just, here's a substrate and here's a product, oh, and here's our ATP hydrolysis to give us ADP, and now we do our calculations and figure out what the free energy change is. Again, with these particular types of calculations, we're concerned about what are the substrates, what are the products, and how much there are. We're not necessarily, at this stage, in these calculations, right, talking about what actually happens. So to go from glutamate to glutamine is actually kind of tricky. We're going to use ATP and actually phosphorylate the glutamate to make an enzyme bound glutamal phosphate intermediate, and then we bring in the ammonia over here, this blue molecule, bump off the inorganic phosphate to give us glutamine. So it's actually much more complicated than what we've said in terms of just, hey, we're going for substrate, 2 product, and we're going to use some ATP and we're going to do some calculations. Okay, so again, when we're simply doing these mathematical calculations of what is the free energy change, we need the substrates, the products, including any ATP or other molecules helping provide some energy, and the amounts of each. And so I did get an email about the extra questions to practice some of this, and I had actually put it on the same form as with Module 1, so this weekend I actually copied that over. So if you look in Module 3, that form is there again, and look at, I think, like the second half of it, it has some more practice problems. We went through a whole bunch of definitions and a few different equations and a bunch of concepts and everything in one lecture, but I think once you actually do some of the problems and plug things into these equations and think about how does free